So I'll start again by repeating my apology that I'm not Laura, and as I said, only a few people here know her, but among many qualities, she is perhaps the best lecturer I've ever met in terms of explaining some of the concepts in dynamic energy budget theory, and it would be great for us all to hear her. I benefited hugely from interaction with her during approaching 4 years when she was a postdoc in my lab, and anyway, it's unfortunate she couldn't be here, but I'll try to cover some of the material that she was going to cover. She and I have had 2 long conversations over the weekend, and we've both worked in different ways in it. The talk contains a mix of material from her and from me, so it's not quite the talk she would have given, but it's got a lot of overlap. And the title is as you see here. And I know the theme was presented as ecotoxicology, but I chose to organize this around a component, which is ecological risk assessment, because it illustrates what I think is the prime reason for using, in ecotox, for using ecological risk assessment, which is that ecological risk involves predicting effects on populations, communities, and ecosystems, but the data that we use typically comes from lab experiments, standardized toxicity tests, and increasingly from suborganismal measurements, including the growing body of high throughput data, which is largely on unicellular organisms or very, very small complete organisms like zebrafish embryos, or commonly single cells. And this trend is clearly increasing. In short, there's a disconnect between data and protection targets, or if you prefer to put it, there's a disconnect between what we know and what we care about. And one of the primary ways in which modelers can contribute is by helping bridge this gap. Obviously, there are a number of different approaches, especially statistical approaches for this, but here I'm going to emphasize process-based dynamic models, specifically those that invoke bioenergetics as a way of making these connections. So why is it hard to make predictions across levels of organization? And I think there are 2 primary reasons for this. One is that at every level there are feedbacks. Within an organism, there are all kinds of regulatory processes. In fact, we already heard yesterday several mentions of homeostasis. There are interactions with the physical-chemical environment, and that came up briefly in some of the discussion at the end of yesterday afternoon. There are ecological level interactions, resource limitation, mutualism, and so on, which, again, we've been talking about here. There is adaptation. Organisms adapt to stressed environments, and there are many documented examples of that. And there are so-called emergent properties. When you shift from thinking at one level to the next level, you can see emergent properties, just as, for instance, happens when you go from molecules to macroscopic descriptions. And an example of that in ecotox would be that the combination of feedbacks within an organism can sometimes lead to, well, from a mathematical perspective, to bifurcations, from an interpretation perspective to tipping points. And all these things say we're going to need models with nonlinearities to make these connections. This workshop takes place against the background of a previous Nimbus investigative workshop on a similar topic, but also on 2 previous Nimbus working groups, smaller working groups that aimed to transcend the levels of organization. One which was led by Cheryl Murphy, who, if you haven't met her, is sitting in the back row here, and myself focused on the suborganismal processes connect, and Angie was also a member of that group, and Phil, who's hiding in the corner there, was also a member of that group, this was focused on suborganismal processes, and a second working group led by Valerie Forbes and Chris Solis looked at transitions from organism to ecosystem and even ecosystem service levels. And I've put the URLs for these on the slide, but if you just go to the Nimbus webpage, you'll find the descriptions of these groups fairly painlessly. Dynamic energy budget models, which I'll describe more carefully in just a couple of minutes, focus on the level of the individual organism. But I've also mentioned that we're concerned in Ecotox, we have to think about how to connect to lower levels of biological organization. And broadly speaking, there are two increasingly used approaches. The first, which I'll talk about, is dynamic energy budget modeling. And you can think of this as a top-down approach that essentially reverse engineers an organism. It's a good approach for thinking about the information you would get from standardized toxicity tests, because you start at the level of the whole organism and you say, if the response to exposure on growth is this, and on reproduction is that, and on respiration is that, what must be going on inside the organism? When I was a physics student, I got dreaded black box experiments where you had a box with, let's say, 4 terminals on it and a couple of measuring devices, and the test was to say what's in there. And to some extent, DEB modeling, at least historically, has taken this approach. And the alternative, which is widely used in in fact, is substantially more used currently, is the identification of so-called quantitative adverse outcome pathways. And this, in many ways, is a bottom-up approach that describes, and I'm here quoting from possibly one of the defining papers by Gary Ankley and others, the linkage between a direct molecular initiating event and an adverse outcome at a biological level of organization that is relevant to risk assessment. Both approaches are shortcuts in it, and each has weaknesses. The DEB approach, as I'll show, it actually gets very fuzzy and imprecise when you try to talk about what's happening at the level of, at suborganismal level. There's ambiguities. And the definition of AOPs given by Ankley is great in principle, but actually once you get up the causality chain, the connection with endpoints that relate to growth and reproduction and survival gets weaker and weaker. So against that background, here's the outline of the talk. I'm going to start by saying something about what dynamic energy budget theory is, and then I'm going to say something specific about how it has been used in ecotoxicology. Since much of the interest is in higher levels of organization, I'm going to say a little bit about the connections between individuals and connecting individuals to populations, because a lot of people here are working directly from a population level description. I'm then going to say something more specific about the way you can incorporate some of the issues in ecological stoichiometry that are the reason that we're here into the dynamic energy budget framework. And then I'm going to end with 3 items that Laura and I picked up as being directions in which we thought there was scope for a further use of a dynamic energy budget theory. In brief, the first 3 sections are targeted at people here who are unfamiliar with a dynamic energy budget theory, and they're a kind of superficial run through a bunch of what exists. And this will be quite fast. I'm a bit unapologetic about the fact that it's fast because I want to leave time for the last 2 sections, and in particular the last section, which hopefully opens the way to some discussion. So let me start by saying something about what dynamic energy budget theory is. And I straight away mentioned that the term dynamic energy budget theory is actually used in the literature in two ways. To me, the obvious way is dynamic energy budget theory deals with dynamic models of energy budgets. That doesn't sound like rocket science. But in fact, that's a very broad definition. It's actually the one I like, but there's a particular body of well-developed theory, which is essentially a theory of metabolic organization that was put together largely by Bas Kooijman and a Dutch theoretical biologist and people working with him. And in a sense, that has captured the name. And so often when people talk about DEB theory, they specifically mean Kooijman's flavor of DEB theory. And to some extent, that's fair because it's by far the best developed. But I'm reminded of a situation that prevailed in prehistory when I was an undergraduate, where the term applied mathematics meant fluid dynamics, and again, one group of people had hijacked a name, and if you were working in mathematical biology or bioeconomics, it wasn't applied math. At least it wasn't what people thought of as applied mathematics. So what are DEB models? They describe the assimilation and growth of energy and elemental matter by living organisms, and in the context of stoichiometry, the elemental matter here matters. At the core of dynamic energy budget theory is a model in which an organism's growth, reproduction, or mortality are related to food intake and environmental conditions by a set of dynamic equations. Actually, I've given an excessively narrow definition there. That's how it applies to an animal. The theory actually has much broader applicability. There's a lot of work on microorganisms, a not so large but still significant amount of work on plants and so on, for which that definition obviously needs a slight modification. The transition from individual to population occurs through using either structured population models, in which a few people here have expertise and some have way more expertise than I have, and or on using individual-based population models, and I'll say just a little bit about these. At this point, I simply want to emphasize that both structured population models and individual population models actually exploit functions and parameters that are properties of the individual. So they're actually not as different as you think, and I'll say a little bit more about that. In a minute, I'm going to briefly describe Bas Kooijman's so-called standard model, which describes all life stages of an organism and involves, in its fullest form, half a dozen individual, what we call individual or I-state variables, and around a dozen parameters. And I'll say again a bit why there is a wiggle room around both these numbers. A lot of the models that are more widely used, including for example, the model that Angie talked about in that very nice little short presentation yesterday afternoon, are really biomass-based model in which there is one variable that characterizes the organism, effectively its size, and in that model case, it's aggregated over the whole population. And a lot of what I'm talking about has its counterpart in biomass-based models, and I want to just highlight this book in case, for anyone that hasn't seen it, by Andrew DeRose and Leonard Persson on a broad range. It's essentially about it's a broad survey of population and community dynamics but that's in models that take specific account of ontogeny. So that's a useful reference. And before I present the model, I want to make a comment about the jargon used by people who work in DEB theory. My personal opinion is that bad choices of jargon have greatly reduced the extent to which this theory is used. And I like to do this by a quotation from Through the Looking Glass. Appropriate to do it at Nimbus because the author, known to us as Lewis Carroll, was actually a very impressive Victorian-era mathematician. But in this, Humpty Dumpty says, when I use a word, it means just what I choose to mean, neither more nor less. And in dynamic energy budget theory, if you use Kuyman's book, you make that substitution in the quote. And specifically, there are concepts like structure, reserve, and some others in DEB theory that actually don't mean what that don't mean what everybody else means by these terms. And my plea for this is that when we get into discussion of this, we try to duck these issues of jargon. So with this warning, let me just show you fairly quickly what the so-called standard model of growth and reproduction as developed over the years by Kuyman is. The idea is that, I laid the pointer down somewhere, oops, what have I just done? A pointer. The idea is that there is food in the environment, the animal feeds, some fraction is assimilated, the rest goes out to species. It's assumed that initially it goes into some compartment called reserve, and that again is different from what most of us mean by reserve. And then it's partitioned, and some of it is used for growth and maintenance, some of it is used to support increasing maturity, which is developmental costs in embryos and juveniles, and to support reproduction among adults. And there are equations that describe this. If we want a model to be transferable to population dynamics, we also have to think about mortality. And of course, bioenergetics is only part of mortality, and for many natural populations, it's actually potentially a rather small part. Predation, disease, and so on are all potentially important. But DEB theory has a rather nice characterization of the physiological component of mortality, and it's a generalization of the idea that of the ideas that are built around the idea that oxidative damage is a component, is a key component of aging and eventually mortality, which is a controversial idea, but the abstraction is that essentially the faster animals are metabolizing and turning stuff over, the more something that we can call damage occurs, and you then have information that takes the production of short-lived ROS species. This leads to damaged proteins, membranes, and DNA that in turn relates to mortality. There is one representation of this in Bas Kuyman's book. At the risk of advertising my own group, I would point out that Tin Planschek, Eric Muller, and I have generalized this, and there's a paper out there that came out just over a year ago. It's relevant for ecotox because, again, oxidative stress is, of course, one important mechanism in the context of the damage of the damage that exposure can cause. Mathematically, what does Kuyman's model look like? There are 4 variables here that correspond to the boxes in that picture that I showed, and then there are 2 variables, if you want them, that characterize the mortality component. Often the model is studied without these, and again you have a wiggle room in terms of the numbers of parameters. And the model has a rather powerful feature, which is that you can characterize the parameters as those that ought to be broadly invariant across species and those that will vary in a systematic way with size. I'm not going to say anything more about that in this talk, but it provides an alternative to the theories out there in the metabolic theory of ecology papers, for instance, to explain inter-specific body size scaling relations. I just mentioned that that is sitting out there. And I've listed, I presume that we can post the slides after this. I've listed various references. I would point out in particular that there is a review of dynamic energy budget theory by a large group of authors led by Marko Jusup that came out early in 2017. And that has a bunch of comments after it that are potentially quite useful. When you put this together from the boxes and arrows in the diagram, you get a bunch of balance equations, and you then have to write down a bunch of functions. And for our purposes, all that matters is that you get a collection of ordinary differential equations. And I'm not going to talk any more about the specifics of the equations. I mentioned ambiguity in jargon, and there is and that leads to the fact that there is actually a challenge in relating the variables to the real world. Laura likes to call everything that the Deb theory handles the fantasy world, and in the fantasy world there are forcing functions like food quantity, food quality, temperature, and so on. These in turn influence ordinary differential equations. You have state variables, and the ODEs tell us what's happening over time to the state variables. But they are these ill-defined things called reserve and structure. And you actually need a bunch of auxiliary equations that connect the abstract quantities with anything that you can measure or observe. And this is the link that makes connections to data. One of the challenges is that for this reason, it's very hard to test a DEB model, because either you've got when you get a mismatch of data and a observation, either the model equations are wrong or your auxiliary equations are wrong. And I put that out there as just something that needs to be thought about. It sometimes leaves you with the impression that people who work with Deb models are pretty weaselly, because you say their model doesn't match the data and they say, well, and they do make a bit of change at that level. It's something to bear in mind. And that's just a repetition of the same slide. Based on what I've just shown, perhaps for anyone that wants to use it, one of the more important pieces of information is that there is a huge data repository of parameters for dynamic energy budget models called AddMyPet. And it's got that name because users are encouraged to parameterize using their processes, their models, and put up the data. It used to be informal and coordinated by Bas Kooijman. There's now a group of coordinators, of whom the person for whom I'm substituting, Laura, is one. There she is. And there's a summary there of what each entry contains. As of January of this year, according to Laura, there were 1,012 species from 120 authors. All what they call large phyla are covered in there with greater or less degrees of precision. Some of the estimates are very thorough and rigorous, and there are measures of quality there, and there's a range of organismal size of body mass across 16 orders of magnitude. So it's an important resource to be available, to be aware of. How are dynamic energy budget models used in ecotoxicology? Well, I just said the content of the first bullet point, what they are. The key to making progress here is that you actually couple it with a toxicokinetic and a toxicodynamic model represented either explicitly or implicitly. Toxicokinetic models describe the essentially intake and output of intake, output, and transformation of contaminants in an organism. Toxicodynamic models characterize the interaction between the internal concentration of the toxicant and the animal itself. The toxicokinetic model could be implicit. You could simply say internal concentration is proportional to external and a pseudo-equilibrium assumption, but it can be explicit. The link to population dynamics, and I'll show an example in just a minute of this, is in principle made by individual-based models, but an approximation to individual-based models that opens the way to more mathematics are structured population models. And at the very end, one of the challenges we'll talk about is whether this can be done for ecosystem processes. If you're interested in taking this further, I strongly recommend an ebook published by Chanling Yager, and it's available from the URL listed here. It's another beautifully clear exposition of some of the stuff I'm rushing through. Oops, wrong computer. So how do we actually implement this connection of between dynamic energy budget models and ecotox? The key is this idea of physiological mode of action. In the DEB model, there are a bunch of processes. There's feeding, there's mobilization of reserve, there's maintenance, there's partitioning, and there's egg production. In DEB, in all the early DEB applications, these are characterized as physiological modes of action, and any one of them could be used as the model description of what a contaminant does once it's inside the organism. And this opens the way, and this is what our Nimbus working group has been largely doing, to making a connection with the adverse outcome pathway paradigm. This picture is an adaptation from a picture in the Ankley paper, I think originally done by Cheryl and transferred among so many of us in the working group that I've no idea how many mutations this slide has been through, but it describes the key thing, and you have chemical processes in the environment, you have some molecular initiating event, and then you identify a bunch of key steps along the chain of causality, which, not surprisingly, are called key events, and this leads to some kind of adverse outcome. I should have said at the start, and I'll say here when I see this, that it reflects an ambition that is shared with DEB modeling to have descriptions of toxicity that are chemical agnostic. That's to say, any chemical that produces the same molecular initiating event is supposed to produce the same subsequent effects, and that's part of the reason that it's had this effect. And the connection that I think can be made and makes a lot of this work relevant is that the idea is that you can relate key events in the AOP with the physiological modes of action in DEB model. And this is especially appealing because work that I don't have time to describe shows that this reverse engineering process often gives you ambiguous results. And it's good to be able to bring a mechanism from the real world rather than the fantasy world into pulling these things together. And, again doing it in the wrong computer. And this slide, which anyone who was at CTAC last year may recall, illustrates an example, again, from the Nimbus working group. This is a characterization of an AOP associated with an endocrine disruptor that is relevant for some fish, including trout, and Cheryl and Dina Lika in Greece and a number of others of us in the group are working in a study that takes the DEB model and then couples it to a simplified quantitative characterization of the dynamic energy budget model. And I'm showing this to emphasize that it actually is possible to take this DEB framework and connect it with a lot of mainstream thinking in ecotox and come up with hybrid models that I'm hoping might have some predictive power. I presented all this as a very rushed through, this is really the first two parts of the talk, a rush through the background of what's there in order to lay the ground for something that makes a connection with a number of people here, which is how to make the connection from individual to population. And at this point, I want to make a step back and go through a very simple couple of steps of reasoning that I hope help make some of the connection clear. Those of you that experienced an introductory ecology class at some point in your misspent youth probably got some lectures on population dynamics that would start with life tables, where you knew a fecundity schedule, a survival schedule, and you calculated things like lifetime reproduction, intrinsic rate of growth, and so on. And let me remind you how that argument goes. You have a population, you divide it into discrete age classes. There's some fraction that survive to a given age. There's a number of offspring. Then the average number of female offspring per female is given by this sum. Essentially, it's a weighted sum of the fecundities associated with all the particular ages. And this is just a snide remark that it's almost always called net reproduction rate in the textbooks, even though it's not a rate. And in continuous time, the analog of that sum is that. And the reason I'm mentioning all this is that using the standard DEB model for any given environment, we can actually solve and get the beta functions and the S functions, and we integrate it, which says that we straight away can get a measure of net reproductive rate from a standard DEB model, and we can then look at how it changes in response to contaminants. And this, or its counterpart for little r, which is the exponential growth rate, has been done for probably around 30 years by, again, in the mainstream ecotox work. As again, we learn right at the start of our ecological studies, this kind of approach has very limited applicability because in the real world, as I've already said, there are feedbacks. The individuals in an organism eat food, that changes the food density, and so on. And now there are a number of different approaches. The models like the model that Angie talked about yesterday, can be derived from properties of individuals if we make the assumption of what the DeRose book calls ontogenetic symmetry, and that effectively says that all physiological rates for individuals scale with biomass or scale in the same way at least. And the most limiting assumption probably is that all our, oops, I did not mean to go back, I meant to do the pointer. All organisms experience the same risk of mortality, and you can couple these with an ordinary differential equation describing the environment, and this is the individual-based characterization of these biomass-based models that I missed yesterday morning, thanks to the weather, but I'm sure some of what you heard then was, and it was certainly true in the afternoon session. Relevant to what Angie is planning as one of the breakout groups in this meeting is the fact that if you relax the most challenging assumption that the per capita rate of mortality is the same for all individuals within a stage, across lifetime, and say that it's only true within a stage, you can get delay differential characterizations of the population dynamics. And in particular, you can get delay differential equations with dynamically varying time delays. And I think Angie's going to say, I hope you're going to say more about that later this morning. Again, this is talked about quite a lot in the De Roos Person book. I actually have a book chapter which I'm happy to post that specifically talks about some of this. And there's a book by Bill Murdoch, Sherry Briggs, and myself that deals with applications of some of these approaches. In particular, there's a lot on this. So there's background involving delayed differential equations, and it's on both, and there's a corresponding huge background in which, again, there are people, I'm looking at the back here, there's people with more expertise than me that can be drawn on. I just want to show one example where the dynamically varying time delay is informative. And this was some work in the lab of Ed Macaulay in a bit over 10 years ago. He did a bunch of experiments in large tanks on Daphnia and discovered evidence of two different attractors. The population either settled into what we called large amplitude cycles, which are just like the prey-predator cycles in the Rosenzweig-MacArthur model that, again, Angie mentioned, or you got a very different type of population cycle that had a small amplitude. These differed by the fact that the typical juvenile development time, these are experimental data on with the low amplitude cycle and with the large amplitude cycle, and with the small amplitude cycle was very different. These individuals took forever to mature to be adults, and that was the histogram of what was these are from a bunch of stochastic third try. These are from a bunch of experiments on individuals drawn from these populations. These are from the populations themselves, and well, that and that were from the individuals. We put together a delay differential equation model that involved bioenergetics, but not no further Deb subtleties, and the model had the property that you could produce both types of dynamics with Daphnia realistic parameters, and you can go all the way and do some of the math. You can produce bifurcation diagram with a set of parameters here. The algal carrying capacity is the bifurcation parameter, and this shows you can have a stable equilibrium, an unstable equilibrium. From the unstable equilibrium, these blue lines represented the maximum and minimum population for biomass for the algae. This represented it for the on the small amplitude cycles. This represents it for the large amplitude cycles. So you can bring to bear the conventional mathematical tools for which there's a lot of software and so on that can help us. But there's another approach for which Deb is particularly well suited, and that is using individual-based models. And I emphasize that these use the same information as the structured population models. And again, at the risk of self-advertisement, I've put up here a pointer to a paper that three of us published in ecological modeling in an issue that was honoring the work of Don DeAngelis, who has worked in both areas, that came out just about a year ago, that emphasizes the complementary insights you get from these and the power of working with both of them at the same time. And the background was that Ben and I worked with Andre. We used an individual-based model. Andre had previously published a structured population model. We both learned new things from the other's approach, and you can follow that up. Specifically for Deb, there is a paper published with code for so-called Deb IBM, would let you take the Deb model and implement it in the NetLogos software package, which is free, well documented, and so on, and make predictions of population dynamics. And one of the striking things that Ben Martin did when he was a PhD student in Volker Grimm's lab in Germany was he did a study on chemostat experiments on Daphnia magna, a different species from the one Ed had done. He took the parameters from that. He then tried to describe experiments like those that Ed Macaulay had done earlier, where you got the two types of cycle. This model, of course, has stochasticity in it, the individual-based representation, and he got jumps between the two types of cycle, and he got the developmental delay changing to being large when the small amplitude cycles were running and small when the large ones were going. I put that in to emphasize the congruence of the approaches and the different kinds of insight that you can get from them. Ben Martin also applied this to ecotox, the study of an insecticide on a contaminant that appeared to have primarily an effect on reproduction, and he got a good agreement of a model and data. But he then used it to get an insight that perhaps helps us understand why some of this DEB stuff is important. Because after trying to match experiments, he then went back and said, what are all the different physiological modes of action that could occur according to our DEB ideas? And just like I showed before, it could be here, 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 or here. There's a bunch of different places. He then ran the individual-based model for a series of situations that would lead to the same result in a standard OECD-style chronic toxicity test, a chronic Daphnia reproduction test. Depending on which mode of action was developed, you either got parallel decreases in biomass and in population abundance, or you got a decrease in abundance with no change in total biomass, or increasingly, you got overcompensation in numbers, an increase in biomass, increase in numbers corresponding to a decrease in biomass. In short, the suborganismal assumptions we make matter if we care about the differences between numbers and biomass. And if you're now thinking about real Daphnia in a real, or real zooplankters generally perhaps in a real system, size matters because size relates vulnerability to predation among a whole bunch of things. It affects total secondary production. So now, how do we make the connection with stoichiometry? These abstract reserve and structure quantities that I've talked about, actually we can think of as characterized by so-called generalized compounds. And they are some mixture of carbon, nitrogen, oxygen, and hydrogen, for example, but we can add elements as we see fit. DEB assumes that the composition of reserve and the composition of structure is invariant. You can write the chemical formula for the compounds in this form, where you always set the C to the carbon level to 1, and that gives you a measure of biomass in what are sometimes called C moles, and this is actually a very nice unit of currency in a bunch of these things. And if you want to figure out whether you understand C moles, just try to figure out how many C moles there are in a kilogram of glucose. If you could do that if you could do that little example, you can do it anywhere. Let me remind you what the model looked like. We had these components: reserve, structure, reproduction, and so on, and X for food. And now let's write down what the formula look like. These are each of the components. Each of them has an elemental composition. If the flows are as described by the ordinary differential equations that I've described, and we want mass balance, there are going to be mineral products that we have to assume in the model are excreted by the organism. There's, for example, CO2. There's water. If we're dealing with an animal, an aerobic animal, actually it's an input of O2 here, and there's certainly going to be nitrogenous waste. And we can extend this to cases like we heard about yesterday with phosphorus. We could just add phosphorus, but there is then going to be another mineral associated. And actually, I was going to make a remark here. Let me make that remark at the end. But there are, in each case, some problems with this characterization. My point is that here is that to get mass balance, you have to consider not just the processes going on within the organism. You have to consider the interactions with the environment. DEB does allow us to have in spite of the fact that the individual compartments have fixed stoichiometry, DEB does allow for variations in stoichiometry within a compartment, provided the chemical formula for structure and the chemical formula for reserve are different one from another. Then if you change the ratio of reserve to structure, you can change the ratio. And for example, if we, loosely speaking, imagine that there is more carbon in reserve and more nitrogen in structure, if we're thinking protein, that the structure has a higher proportion of protein, then reducing the reserve ratio gives you a change in C to N, as illustrated here. If you actually want to derive the equations for these balances, you actually have to write down some chemical equations. And these are commonly referred to as macrochemical reactions. And I'm writing them initially here as unbalanced, that if there is some energy required in the process of assimilation, food with an input of oxygen gives you a whole bunch of both organic and mineral products. Likewise, the growth process, likewise, the dissipation. But we learnt at high school and in introductory chemistry, if we had to endure such a class, that you need to balance your equations. So every equation here has some coefficient in front of it. And the point is that as long as you balance the equations, these are not all independent parameters. I'm going to skip the next slide. I just want to mention that there is a concept that is widely used in DEB that lets us bring to, imagine how two fluxes come together, the so-called synthesizing unit. You can think of it as a generalized enzyme. It follows similar principles to Michaelis-Menten, only it carries it over to multiple substrate. And this slide just describes all the possible transformations. You get a bunch of equations and you get the pseudo-steady state, just like you did in the derivation of Michaelis-Menten. And likewise, I want to very briefly run through show you just an example where the SU concept could be used briefly, and it's an example where stoichiometry is, in a sense, the key, and that is for what you could describe as a superorganism like a coral, which involves the symbiotic relationship between internal dinoflagellates that produce energy from photosynthesis and an animal that feeds. And Eric Muller, a number of years back, put together a model where you had and this is just the key steps of it, a situation with an animal coupled to an alga. Each exchanged the surplus mineral that it had described, and the model was capable of describing a lot of features associated with coral biology, and could be extended in various ways. And a postdoc, Ross Cunning, in the past couple of years has extended that a lot, and I'm happy to talk more to anyone that wants it. Because I want to now, against all that background, I've really rushed you through a lot of background because I want to present some some key challenges. To both Laura and me, if you're going to work on a ecological stoichiometry applied in areas like the ones we're applying, we have to have ways of taking advantage of data on stable isotopes, because so much of the information about the flows come from there. And the classic simplification for much of the existing theory on stable isotopes, including the theory on fractionation, assumes that essentially some compartment within the organism, let's say whatever tissue it's taken from, is just a bag of let's say nitrogen atoms across the two isotopes. That's of course not true. What there really is a bunch of small molecules and macromolecules, each containing nitrogen, from which the sampling takes place. And in Deb theory, it would come from these generalized compounds. So Deb theory actually recognizes that, or gives us a way to recognize that stable isotope fractionation and even stable isotope flows are not necessarily described in a way as simplistic as this. And Laura, when she was a postdoc, developed some very beautiful but very complicated theory for handling this situation. And she recognized that there had to be incorporation of what she calls molecular shuffling, which I'll describe in just a minute, and what she called molecule selection. In other words, it's recognizing that there is more going on than just random sampling from this bag of atoms up here. And she pointed, when we were having our conversation, she pointed to a very nice example that showed you had to think quite carefully about the reshuffling. This equation here is the standard way of writing down the macrochemical equation for photosynthesis. And if you just imagined that as what was going on, you would imagine that the O2 that was produced involved oxygen coming from CO2 and oxygen coming from water. In fact, labeled experiments show that all the atoms of O2 actually come from water in that. So we actually have to write down the macrochemical equation not in its simplest form, but in a way that takes account of the fact that we know some biochemistry, or some people know some biochemistry. And Laura's written down the change that takes place here. Note you put 12 H2Os on this side and 6 there. You would normally just cancel these out. But because we're now because we have to extend this now to involve the different isotopes, we actually have to take this account. In terms of this process that she called molecular selection, you have to recognize that Deb doesn't take account of the fact that when reserve, as it's called, is mobilized, it can be used for catabolic processes or for anabolic, that is, biosynthesis processes. And again, there are differences. A big component of maintenance is the turnover of biomass. And in the Deb model, we actually don't care, in a sense, whether we pay our taxes first and don't worry about turnover of biomass, or whether we convert it into biomass and turn it over. As soon as you're dealing with stable isotopes, this really matters because it tells you the fate of the isotopes within the organism. Laura has written this down. It involves complex notation, additional parameters, and it's desperately screaming out for somebody to simplify it. But the principles are defined here. So that's the first challenge that we thought was important. The second challenge, which Laura mentioned to me, is making connections with some of the more mainstream ideas in nutritional ecology. And I'm embarrassed to say that the paper she told me about, which is this paper, and I think we have one of the authors, we have one of the authors here, I had not read it until I read it in a rush on Friday after my conversation with Laura. But the idea here is that, the idea in that paper, as I understand it, and I would love to be corrected, is that there is a lot of overlap, especially related to concepts of homeostasis, between what's come to be called nutritional geometry and ecological stoichiometry as we understand it here. And the ideas behind nutritional geometry had their origins in studies of insects where animals were moving around with choice of food items and trying to achieve some kind of balance, whereas much of Deb theory and much of the current work on ecological stoichiometry was very much influenced, in fact probably over-influenced by work on organisms like Daphnia that were indiscriminate filter feeders. But in both cases there was target work involving achieving some kind of homeostasis within the organism. And this paper, which I think it's fair to say only came out a few weeks ago, is it, or a month or two ago, I'm not quite sure, but this paper points to a potential unification and makes a connection with what are sometimes called multi-reserve DEB models. And what I mean by multi-reserve DEB models are models like, for instance, a model that takes account of multiple food types, multiple reserves, but one type of structure. And there is an example of that actually in a very old paper published by Kuiper and a couple of others, including Bas Kooijman, which was actually a copepod model that was put together. And this model, which I remember recoiling from because of its complexity, for I did read that paper, broadly speaking has a box and arrow diagram that looks like this. It's fuzzy and I'm not worried about that, for I'm not expecting you to absorb detail. Just note that there's two types of reserve, there's a bunch of synthesizing units, and there's some organismal level endpoints. This model was rewritten in the Sperfield paper like that, and it shows the power and the seduction, as it were, in different ways of box and arrow diagrams, that scheme is pretty much the same as that scheme, set out differently and with different representations. This is clearly something that can be taken further, and if we move away from what I think is sometimes called the algae Daphnia fish axis, if you like, that perhaps is too dominant in a lot of ecotox, and I'm certainly guilty of that myself. We want to make connections with this body of work, and that is I think important. And the final challenge that I want to put out there is that we need to better connect with ecosystem processes. And one of the aims of the parallel working group to the one Cheryl and I were handling was to take one of the standard DEB models, standard bioenergetic-based models that was out there. They chose one called Ecotox, and to try and incorporate a DEB characterization of some of the organisms within Ecotox. The reason that proved, or one of many reasons, other than just a sheer labor intensiveness of the work, that proved difficult, related to these mineral products that I'm describing. If you're thinking of an ecosystem, it's not just that, say, phosphorus is excreted, it actually matters what form it's excreted in, because if there's a inorganic phosphorus excreted by an animal, it can relatively quickly be used to support further algal growth. If there is an organic form produced, it requires microbial action for a subsequent transformation and availability, and so on. In other words, it's necessary to couple together the DEB characterization with some of the concepts from the fantasy world that ecosystem ecologists use. There's actually two fantasy worlds there. I've been talking about a fantasy world simplifying what organisms do, but the ecosystem people talk about dissolved inorganic nitrogen, dissolved organic phosphorus, and so on and so on. These are just such oversimplifications. They're essential for modeling, and yet we somehow have to, each, in a sense, ignores the complexities of the other, and it somehow has to come together. So, in short, I've tried to give you a very quick run through a whole lot of different areas the DEB theory covers. I've pointed to what Laura and I think are potentially three important areas ripe for further development. You may have others, so thank you for your time.
So, I have many questions, but can you go back to the DEB? They have this kind of scheme, you have this kind of diagram.
Oh yes, just any of the ones with the scheme in it. Oops, what have I done? Oh, Actually, let me go to the slides order. Oh, why did it? So you're thinking of something like that one?
Oh, yeah, that's fine. That's perfect, yes, thank you.
Okay, let me just leave it.
So I assume this scheme actually applied to individual, let's say.
Yes.
Okay.
Yes.
But when an individual, let's say, you can have different stages, right? You have a juvenile stage, you have adult. So when you're doing this kind of chi or 1 minus chi, actually itself is changing over time.
Right. So.
I mean, in general, how do you?
How do I better characterize this here?
Because itself is changing. Maybe I don't know. Maybe nonlinear.
Right.
Or maybe
I think
it's complicated. It can be.
This is perhaps one of the more controversial bits in the standard form of the theory, and I think in part it's another case where the partitioning is, where the controversy is overstated. That the important point here is that if I forget about this term, which is often quite small, the partitioning is to growth and maintenance, and on the one hand, and let's talk about an adult, to reproduction on the other. Now, as an organism grows, for instance, if it's exhibiting von Bertalanffy growth, then the maintenance costs increase faster with size than does intake, and hence mobilization. And so what you get is a steady decrease in the rate of growth. Sorry, I've got the two. I've written I got the diagram the wrong way around. It's this one I'm ignoring. This is growth and maintenance, and I get decelerating growth with more and more of this going to maintenance, and a sustained rate of reproduction, and I get that even with a constant value of kappa. If you now simply replot these results the way it's often done in in the literature, where you, instead of looking at this somewhat abstract quantity which relates to that, you look at net production, which is assimilation rate minus respiration. What this gives you is actually a size-dependent allocation to reproduction, which is very similar to what is found in a lot of data. I once tried comparing the two sets of predictions for some very detailed Daphnia data, and the answer was both did just fine in describing the data. The bit that is controversial, and I think was at the heart of your question, is that if you now contrast juveniles to adults, juveniles are presumably assigning nothing to reproduction, and then part of that is covered by an idea that to me makes sense in embryos and very young juveniles, but then becomes hard to defend, which is that there are high energetic costs associated with increase in developmental sophistication, development of an immune system, et cetera, various costs. But nonetheless, the problem you describe is there, and I think there are two ways around it. One is to which I think is the one that Bas Kooijman, if he was giving this talk, or Laura, if she was giving the talk, would probably say, is that actually what this describes is the allocation of energy to material destined for reproduction, and you actually need another module attached to this, which is egg production, and that's actually what happens in the model that Cheryl and Dina are working on for the trout. And the other, well, there are various other defenses, but you've hit on a weakness that I think is claiming is real but non-fatal, and there are workarounds. But
yeah, I mean, I do like this diagram. It's fabulous,
No.
because I'm kind of connecting to. I've been working with behavior ecology, right? Because in the slides you have been talking about this kind of the insects, right?
Yes.
Especially social insects. Actually, you can consider the whole colony. Actually.
sorry, can you? Is that switched on? I'm finding it difficult to hear you.
Probably just me. This all.
Oh, right.
Okay, so it can be connecting to the, let's say, the social insect colony itself can be
Yes.
considered as a superorganism, right?
Yes.
So they actually do have this kind of, let's say, division of labor. They have different task allocations. Actually, can be very corresponding to what we are doing here.
Yes.
Also, depending on the stage of colony, actually, at the beginning, you can more like everything is more kind of growth, right?
Yes.
And then later on, then you feel mature, and then you can have more kind of allocation for the reproduction. So I'm kind of trying to do this kind of mapping and hoping there's some kind of a connection between the things we have been doing and then this diagram. So another thing I have been kind of have kind of concern, or maybe you already know, or you'll be already doing the modeling on. They have actually involved multi-time scale there. So I don't know how you're doing the modeling. How do you resolve this from food to income and outcome? But in the meantime, you talk about more process there. So I don't know when.
So to me, you've asked three questions there, and I didn't mean that as a negative, but let me try and separate them out. One is making connections with behavior, and that in principle, if we're dealing with non-social insects, for instance, with multiple food types, that is relatively, that in principle is consistent with the framework. We just have to have a much more sophisticated characterization of feeding. In the context of something like social insects living in colonies, even the mainstream literature on that I don't know well, but certainly to my knowledge, nobody has tried. I'm sure people have done some work on the bioenergetics of that, at least I would guess it, but I've no idea. I just plead ignorance on how that would fit in here, but it would be, if you like, it becomes challenge 4 on the
For sure.
here. And you had, but you'd one other point, and
The time scale, but
no, time scale. And time scale is the one that I do actually have the.
People have other questions, but
no, time scale is actually really important, because a lot of a lot, and that's why I did want to say something about it. A lot of the model prediction of this sort is it's very much aggregated behavior. So, for instance, in the rainbow trout, there is energy becomes available actually in a non-linear way over a year for reproduction, and then there is a spawning event. And the model definitely needs tweaking, and in some cases more than tweaking, if you want to take account of shorter timescales. And there's actually a paper led by Eric Muller that again came out of the Nimbus working group that specifically looks at that in the context of trout reproduction. And that's a situation where there is a situation where there is also a quantitative AOP model that for which it's possible to make comparison. The other separation of timescale is separation of timescale of chemical processes, and that was well illustrated, though I glossed over it on the slide on oxidative damage. That the average lifetime of an ROS, reactive oxygen species, in many cells is, I gather, less than a nanosecond, or certainly less than microseconds. You know, it's a very, very rapid. It's a very rapid turnover, and so in fact, in many cases, it's defensible to imagine that you've got a pseudo-equilibrium there, and to work with one equation which represents damage. And again, there's a large group, especially in Europe, who work on mainly survival data, who do exactly that. So all the tools for timescale separation are there, but some of the ones, like the one I talked about in terms of reproduction, the simple separation of timescale arguments don't work, and you actually need to couple in some new assumptions.
Thank you. Appreciate it.
Thank you for that presentation of the DEM model. I know it's not simple. I'm a fan of looking at multiple elements and their concentrations in different organisms beyond carbon, nitrogen, and phosphorus. And I think there's good justification for doing that. We know that copepods in lots of areas of the world's oceans can be limited by iron, for example, right, and things like that. So I'm wondering if, similar to applying the model to other kinds of organisms and populations, are the tools there to incorporate other types of elements? Or do you see already challenges with doing that?
I think in some cases the tools are there. If I chose a different example, for instance, I think I recall somebody doing, and if not, I know it would be in principle straightforward to do some DEB modeling for diatoms where silicon was the key element. But really, that type of thing is a case of having similar concepts, different names of the mathematical variables, and slight difference, and obviously differences in the connection between model and data. Your iron example is more complicated because iron is involved in some of the iron is involved it's not just a matter that it's potentially limited, it's the chemistry that's involved in which goes beyond which goes beyond a lot of my knowledge, but in particular in ecotox context, iron is involved in various toxicity and repair mechanisms. So I admit I'm giving you an embarrassed don't know in the context of iron, but I am saying there's a bunch of other let's say, structural elements that are relatively straightforward. I don't know if that's if that helps.
It does a little, but only in, I guess, pointing out some of the challenges.
Oh, yeah.
Iron,
Okay.
Iron's kind of a bad example but there are other elements like it in that it's a cofactor for enzymes that feed back into things like energy and biomass and changes over time in biomass at the individual and I'm assuming population level. So there are other elements like that, cobalt and copper. But I guess another question that's related to this that might be a potential challenge is that some of these elements and even some of the macronutrients that people were talking about yesterday in their talks is that there's not a linear relationship between exposure or accumulation to that nutrient and growth. There can actually be negative effects at excess nutrients from that nutrient. And I'm wondering how easy is that to capture in this model? Is it kind of built in, or would you have to really change so many different equations to make it work?
I don't think that latter one is a problem. I mean, I presented this talk in the context of Kuyman's so-called standard model because I wanted to have an exemplar. Obviously, when you go to particular situations, there is particular biology, and I think that negative consequences of an increased macronutrient within the organism might emerge. And I'd need to play around with this to answer, but they might emerge purely on the grounds that they lead to increased metabolism, that leads to increased damage through that module. It might need an explicit representation, but I don't think that's a problem. And in the context of ecotox, you have virtually every situation where you have hormesis relates to what you're saying, and there are definitely examples with hormesis that have been modeled in this way.
Thank you.
Yes.
My question is related to data for this. How would you go about collecting? I'm trying to think of some of the experiments that we have running up at the EPA right now, and I'm wondering how we might go about gathering the data to parameterize something like this.
Right. You mean, are you talking about at the toxicity end, or are you talking about?
I guess I guess in this standard model, like in the absence of toxicity, when we've got these, conceptualized reserve areas and things like that, I know there's some challenges related to kind of measuring food intake of individuals in a feeding experiment or something like that, and then also trying to figure out how to actually parameterize how things are being partitioned in there and things like that.
Right.
So I just wonder, in general data sets.
I'm glad you asked that question because it's a bit that I skimmed over, and I think when Laura and I were comparing the talks we were given, because she's been doing so much on that, she would have done that, and she'd have missed out on the individual to population part. That was just a decision we had to make. But it's possible to recast the equations in a way that uses essentially parameter groups that are meaningful for a broad range of organisms, and that's how this collection of data for over 1,000 species occurs. There is a paper that she's one of the authors of that came out in I think it was 2009 or thereabouts, that talks about the different parameters that you can estimate from different types of data. And then the way this package works uses an approach developed by Dina Lika and again others in the DEB community. Neither of these things was I involved in, which I think you could describe as using what I'd call a pseudo-Bayesian or a poor man's Bayesian approach, and that is that because Deb makes predictions about which parameters should be invariant across species, at a given temperature, and which parameter combination should be invariant, which parameters should scale with size, and how they should scale with size. If you have very limited data on on, let's say, growth and reproduction, a maximum size, just a few pieces of data, you can, in principle, you can get low confidence estimates of the parameters by taking some of the others as default values or default starting values, and you then, instead of maximizing a likelihood, or maximizing a residual sum of squares, you maximize some weighted sum of your data and this sort of pseudo data or quasi data that comes from other information. I think this is analogous to using priors in a Bayesian system, and there is also a real Bayesian methodology being developed, but hasn't been used to any greater extent in this. I think the leader in that is Leah Johnson, who she was in Florida, in University of South Florida. I think she's in Virginia Tech now. So that is going. But the key thing is you exploit these interspecific scaling relations, and the ADMAPET database gives you. One of the metrics that it gives you is what it calls completeness of data, and a large number of that means that most of the information from which the parameter estimation derived actually came from your species and the data you provided. A low value for that you can think of as a red flag. It says that a lot of the estimation derived from the pseudo data, the stuff that was put in there. So it's there's a relatively small number of species for which you can get complete complete parameter sets, but this approach, often using, I think I was told often about four of the 12 from assumed to be invariant from another context, give you at least a problem that has a statistical problem that provides a solution, and it's then up to you to determine the extent to which you believe it. So I think, maybe we could maybe talk more about that, but that's that was recognized from early on as the big problem was the parameter estimation, and this idea for which I think the breakthrough was Dina's paper gives you at least a step forward, and there's a lot of documentation in this ADMAPET database.
Okay. Thank you.